All right, so that was the vestibular system. And then we can talk about our last special sense, which is vision. And this is the one that's going to be a little bit more in depth than the other ones. So we'll want to make sure we focus on this one. And we'll definitely cover this in lab as well. So we have 3 tunics that make up the anatomy of the eye. We have our fibrous, or most outermost layer, which is the sclera. And so you can kind of see where this is pointing to. It's this white outside part here, and it has the cornea in it. Then we have our vascular, or middle section, which has the choroid. And this is going to contain our iris as well as our ciliary body here, ciliary body and the iris. And then we have our neural, or innermost layer, which is the retina. And so when it comes to our fibrous tunic, like I said, we have the sclera and the cornea. The sclera is a firm and opaque white layer. And it's just connective tissue, maintains the shape of the eye, helps protect the internal parts of the eye. It's also the attachment point for ocular muscles that help move the eye. And then our cornea, which is continuous with the sclera, is going to be transparent to let light enter the eye. And so you can kind of see here it's a little bit more transparent, helps get light into the eye, and is a protective surface for the iris and lens. Our vascular tunic has that choroid, which is going to be incredibly vascular, which is where all those blood vessels are. Then we have the ciliary body, which is going to be continuous with the choroid here. Muscles going to attach to the lens to help focus that lens on the retina. Focus light coming into the lens on the retina. And then our iris, let's get that color. It's going to be the melanin and colored portion of the eye. So it's going to be maintained with smooth muscle, and it will dilate or constrict the pupil such that we can control how much light is entering the eye. Now our neural tunic is made up of that retina, and that retina has a couple different aspects to it. We have our macula, which is a region of light where it's more concentrated for focusing, and the fovea, where we have the highest visual acuity, the highest focused light centered here on the retina. Then we also have our optic disc, which is a region that only blood vessels are entering out of the eye, and there are no photoreceptors here, so this is also known as our blind spot. And so our neural tunic is made up of these photoreceptors, and then bipolar cells, and then ganglion. So basically the light comes in, meets this retinal pigment epithelium layer, stimulates the cones and rods, which make contact with the bipolar cells, and then those receive those neurotransmitters from the receptors and make contact with the ganglions and carry the neural signal out into the optic nerve. So when we're talking about rods and cones, the rods are definitely more widely distributed. We have 120 million of them, and they're more concentrated on the periphery of the retina, stimulated by dim light, and they're non-color sensitive, and they have the protein called rhodopsin, which responds to that dim light. Cones, we have a lot less of them than rods, 6 to 7 million. They're concentrated in the fovea and the macula, and they are made up of 3 different types of opsins that are sensitive to particular wavelengths of bright light: red, green, and blue. Those 3 cones respond to 3 different wavelengths, and various combinations throughout these wavelengths provide different colors or perceptions of colors that we can have through light. So there isn't a purple cone per se, but if we have a little bit of red and a little bit of blue, we can have purple. Same thing with yellow. A little bit of blue in there, a little bit of green, we get more yellow. If you're missing 1 or 2 of these cones, or they're dysfunctional, you can lead to color blindness in certain areas. And so if you're missing your green or red cones, those will look pretty similar to you in like different shades of gray rather than green and red, very distinct. So I'll go ahead and show another retina pathway here.
See you later, not having Wi-Fi when you want it. Hello to a phone plan with full speed hotspot tethering. Hello Google Fi,
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the retina. The retina contains the neural component of the eye. When light reaches the back of the eye, it enters the cellular layers of the retina. The cells of the retina that detect and respond to light, known as photoreceptors, are located at the very back of the retina. There are 2 types of photoreceptors, rods and cones. Rods allow us to see in dim light, but don't allow for the perception of color. Cones, on the other hand, allow us to perceive color under normal lighting conditions. Throughout most of the retina, rods outnumber cones. In one area called the fovea, however, there are no rods but many cones. The fovea represents the area of our retina that provides our highest acuity vision, and thus is at the center of our gaze. When light hits photoreceptors, it interacts with a molecule called photopigment, which begins a chain of events that serves to propagate the visual signal. The signal is transmitted to cells called bipolar cells, which connect photoreceptors to ganglion cells. Bipolar cells pass the signal on to ganglion cells, which leave the eye in a large cluster at an area called the optic disc. The optic disc doesn't contain any photoreceptors, and so represents an area of the retina that can't process visual information, creating a natural blind spot. However, we normally don't notice our blind spot. The brain uses information from surrounding photoreceptors and the other eye to fill in the gaps in images that are processed by the retina. After leaving the retina, the ganglion cell fibers are called the optic nerve. The optic nerve carries visual information toward the brain to be processed. There are 2 other cell types in the retina that should be mentioned: horizontal and amacrine cells. Horizontal cells receive input from multiple photoreceptor cells. They integrate signaling from different populations of photoreceptor cells, make adjustments to the signals that will be sent to bipolar cells, and regulate activity in photoreceptor cells themselves. Amacrine cells receive signals from bipolar cells and are involved in the regulation and integration of activity in bipolar and...
Okay, so that was the pathway in the retina. And now we can talk about light refraction, which is the bending of light passing from one medium to another. So we have structures in the eye that are more dense than what's in the air. So air is not very dense, and then liquid and solid matter in your eye is more dense. And so that changes the light when it comes in. And so it's literally a different medium. Your eye does what's known as accommodation by contracting the ciliary muscle to thicken the lens when something is close to your eye. And so when something is far, this eye is not accommodating or changing its ciliary muscle and lens for light that's coming in, allowing light to just focus on the back, on the fovea, and send that signal on. And the image of a distant object would be in focus on this retina. Whereas a near object, we have to change the physiology of the eye by accommodating. We contract this ciliary muscle, which ends up thickening the lens. You can see this lens is thicker than this lens. This thickening of the lens bends the light in a different way, such that it is allowed to help focus that light on the fovea again. And this is an accommodation. And so when we're looking at the pathway of how light gets to your brain so that you can recognize it and perceive it, it comes in through the optic nerve, and then it goes to the optic chiasm, optic tract, and then it protrudes out to the thalamus, and then back to the occipital lobe. The way that these are organized are such that things on the right side of the visual field, are coming in, and on the left side of each eye, those are where they're recognized, and that's where the light is actually refracting into. And then those tracks go to the left side of the brain. Same thing with the left side of the brain, or left visual field. Those meet the right side of the eyes based on the way the light is situated here. It's on the left side, but it's meeting the right side of each eye. Those right side of the tracks meet the right side to help you understand and distinguish between what's on the left side and what's on the right side of my body. And so these tracks are crossing each other at the optic chiasm, and then making extensions out to the thalamus, and then going out to the primary visual cortex in our occipital lobe. And so I have one more video for you on the visual pathway.
Welcome to 2-Minute Neuroscience, where I explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the optic nerve. The optic nerve is a sensory nerve, responsible for transmitting information about vision to the brain. The nerve begins in the retina, as the axons of cells called retinal ganglion cells. These axons come together to leave the eye at a region called the optic disc and form the optic nerve. The optic nerve leaves the eye and extends to a structure called the optic chiasm, where it meets the optic nerve from the other eye. At the optic chiasm, the optic nerve fibers carrying information from the sides of the retina closest to the nose cross over to the other side of the brain, while those carrying information from the sides of the retina closest to the temples remain on the side of the brain where they are. After leaving the optic chiasm, the nerve fibers are referred to as the optic tract. Most of the nerve fibers in the optic tract end in the lateral geniculate nucleus of the thalamus, and from there the information will be passed on to the visual cortex. Damage to the optic nerve can occur due to a variety of causes, like trauma, tumors, stroke, or glaucoma. The deficit that occurs after damage depends on where the nerve is damaged, and involves some degree of visual defect or anopsia. If the damage occurs before the optic chiasm, then the patient will experience blindness in the eye supplied by that optic nerve. Damage to the middle of the optic chiasm will cause loss of the lateral visual field of both eyes, due to the way fibers from the nasal side of the retina cross over at this point. If the optic tract is damaged, one half of the visual field will be lost in both eyes.
Alright, so that actually concludes our lecture on special senses. In lab, we're going to dissect a cow eye or sheep eye, depending on what's available, and we'll make sure that we go over this visual pathway as well as the auditory pathway and other pathways if you're interested in staying after and making sure that we're all on the same page. But we'll cover this material in lab as well.